Usually when we think about aging, there's this underlying expectation of it being a totally passive process.
Right, like a car just slowly rusting away in the driveway.
Exactly. Or, the tread wearing off your tires over thousands of miles. You just assume your biological machinery breaks down over time and, well, that's it. It's just wear and tear.
It feels inevitable. I mean, characterizing aging as this slow, passive degeneration is a very comforting, if slightly depressing way to make sense of what happens to our bodies.
But stepping into the world of advanced cellular biology, it completely shatters that simple wear and tear model. We are actually looking at a microscopic landscape that is far more active and, honestly, a lot more shocking.
Oh, absolutely. It's violent down there.
It really is. So today we are completely reframing how you think about aging. We're anchoring this deep dive on a groundbreaking 2026 science paper by Tan and colleagues. And the core thesis here isn't about passive decline. Our organs actually deteriorate because a specific type of cellular trash collector essentially, forgets how to do its job.
Which leads to this localized, highly toxic buildup of what we can really only describe as zombie cells.
Right. It's an intense paper. We have a dense, fascinating stack of source material to unpack together today. The goal is to go deep, leaving no concept, no mechanism, or detail unexplained.
Because we are fundamentally talking about an active failure of a very specific, highly tuned biological system. And to understand this failure, we first have to meet the microscopic cast of characters keeping our organs alive.
The immune cells.
Yes, specifically two main types driving this entire dynamic. We have macrophages and we have neutrophils.
So macrophages are essentially the immune system's primary eaters, right? The cells that clear out debris. But this research draws a really hard line between two different types of macrophages. We have tissue-resident macrophages, or TRMs, and then monocyte-derived macrophages, or MDMs. How do these two actually differ in their day-to-day functions?
Well, the tissue-resident macrophages, the TRMs, are the true locals of your organ tissues. And they have this fascinating origin story. They actually develop prenatally.
Wait, before you're even born?
Exactly. They originate from the embryonic yolk sac. So before you take your very first breath, these cells migrate into your developing organs and permanently set up shop. They make up roughly, 60 to 90% of the total macrophage population in your major organs.
So they're fully integrated into the tissue architecture from day one?
Yes. And they are highly specialized, depending on where they live. So in the brain, we call them microglia. In the liver, they are known as Kupffer cells.
Right, Kupffer cells.
And they live for years. They self-renew locally within the tissue itself without needing any reinforcements of the rest of the body. Biologically, they express very specific markers. These are surface proteins like CX3CR1 and TIM4.
Which kind of acts like an ID badge.
Exactly. It identifies them as these deeply embedded, long-lived guardians of the organ.
Okay, so that's the TRMs. What about the other group, the monocyte-derived macrophages, the MDMs?
MDMs are distinctly postnatal. They were born in your bone marrow and they circulate through your bloodstream as monocytes.
So they're mobile?
Very mobile. They're constantly being generated and replaced, moving into tissues only when there is an acute infection or a sudden injury. They have a remarkably short lifespan compared to the TRMs.
I see. Thinking about this visually, for you listening, the TRMs, the tissue-resident ones, they operate like the lifelong dedicated park rangers of a national park. They know every single inch of the forest ecosystem. They live there, they patrol it, they maintain the trails.
That's a great way to picture it.
And the MDMs, on the other hand, act more like seasonal temp workers or emergency responders. They get boosted in from the bone marrow to handle a sudden surge in tourists or like a forest fire, and then they leave.
Right, the park rangers are responsible for the constant, daily, essential maintenance of the tissue. And their absolute biggest maintenance task brings us to the second character in this dynamic, which is the neutrophils.
The trash that the rangers are constantly having to pick up.
Exactly. And neutrophils are the most abundantly produced immune cells in the human body. The sheer volume is staggering. A healthy person generates between 10 and 100 billion of these cells every single day.
Wait, billions? Daily? That is a massive amount of cellular material moving through the body.
It's a huge energetic cost. They are the first responders of your immune system, designed to swarm bacterial infections. But they are incredibly short-lived. A neutrophil ages and naturally dies within a matter of hours after entering your bloodstream.
Wow, just hours?
Yeah. So because they die so fast and in such massive billion cell quantities, they require a constant aggressive cleanup operation.
Which falls on our park rangers, the TRMs, and this cellular eating process, it's called efferocytosis, right?
That's the technical term, yes. Efferocytosis.
So the TRMs in organs like the liver, the spleen, and the bone marrow, they have to constantly swallow up and digest these dead neutrophils to keep the tissues clean and functional.
And if the TRMs fail to clear them out, the consequences for the organ are disastrous.
Okay, we are about 5 minutes in, so let's do a quick recap. 1, TRMs are your lifelong organ caretakers, the park rangers. 2, neutrophils are short-lived cells born by the billions every day. And 3, TRMs must constantly eat these dead neutrophils to keep the organs clean.
Spot on.
So a question for you listening. Imagine your city's garbage trucks suddenly stopped picking up the daily trash. What happens to the streets after a week? What happens after a month? Keep that visual in mind, because that brings us to what happens in an aging body when the TRMs stop clearing out these billions of dying cells. The trash doesn't just sit there, does it?
No, it doesn't. It fights back. In an aging body, these uncleared neutrophils don't just quietly fade away into the background. When they're left stranded in the tissue, they undergo a really dark biological transformation. They enter a state called senescence.
The zombie state?
Precisely. They refuse to die. The researchers were able to track these senescent cells because they begin to heavily express a surface marker called CXCR4.
So CXCR4 acts almost like a warning flag, or, the glowing red eyes of the zombie neutrophil. It signals that this cell has completely deviated from its normal life cycle.
Yeah, that's a good analogy. And to understand exactly how abnormal these CXCR4 positive cells become, the researchers utilize single-cell RNA sequencing.
Oh, let's unpack that, because that's a crucial tool.
It is. It's a powerful technique that allows scientists to look at the exact genetic instructions being actively read inside an individual cell at a given moment.
So it's not just looking at the DNA they have, but which pages of the instruction manual are currently open.
Exactly. By mapping out the transcriptome this way, they charted the trajectory from a healthy, newly minted neutrophil to a dying one, labeling the sequential states NEU1, NEU2, NEU3, and NEU4.
Okay, so NEU1 through 4. How does that trajectory change when they compared young, healthy mice to aged mice?
In young mice, you primarily see neutrophils in the NEU1 and NEU4 states. That represents healthy, functional cells and then normally dying cells that are ready for clearance.
Makes sense.
But in aged mice, the entire population shifts. The neutrophils get stuck in the middle, in the NEU2 and NEU3 states.
The zombie states. So what exactly is a neutrophil doing when it's stuck in that NEU2 or NEU3 phase that makes it so dangerous to the surrounding organ?
Well, the NEU2 state is characterized by 3 highly destructive features. First, it develops a senescence-associated secretory phenotype, or SASP.
SASP, right.
The cell actively spews out inflammatory alarm signals, creating a constant state of chemical panic in the surrounding tissue. Second, it develops a fierce resistance to apoptosis, meaning it biologically blocks its own natural programmed cell death.
It refuses to die.
Exactly. And third, it triggers cell cycle arrest.
Okay, wait. Here is where I have to push back a bit, because biological rules seem to be breaking down here. Neutrophils are terminally differentiated cells, right?
They are.
They are fully mature, highly specialized, and completely incapable of dividing or reproducing. So how can a cell that cannot divide experience cell cycle arrest? I mean, it shouldn't have active cell cycle mechanics to begin with.
It's the paradox of the terminally differentiated cell. It highlights one of the most surprising and, honestly, sinister findings in this research. You are correct that they don't divide, but cellular biology is incredibly resourceful. These aging neutrophils actually hijack and repurpose the protein machinery normally used for cell division.
Hijack it how?
They take specific proteins called cyclin-dependent kinases or CDKs, and they use them for violent ends.
Wow. So if they aren't using CDKs to split into 2 cells, what are they using them for?
They use a specific kinase called CDK46 to facilitate a process known as NETosis. This is the literal physical breakdown of the neutrophil's own nuclear envelope.
Oh, wow.
Yeah, and simultaneously, they use another kinase, CDK5, to trigger mass degranulation. That's the rapid spitting out of highly toxic corrosive enzymes that were originally meant to digest bacteria.
So they're weaponizing the very gears of cell division to turn themselves into localized toxic bombs.
Sure.
You mentioned NETosis, which sounds like, well, throwing a net. What does that actually look like in the tissue?
NETosis stands for neutrophil extracellular traps. When these senescent neutrophils get stuck and realize they aren't being cleared out, their chromatin, their tightly packed DNA decondenses.
It unravels.
Right. It mixes with those toxic granular enzymes we just talked about, and the cell violently extrudes this mixture into the surrounding tissue, creating a corrosive web-like trap of DNA and protein.
That is horrifying. Going back to our city trash analogy, the uncollected trash bags sitting on the sidewalk aren't just smelling bad. They are now actively sprouting toxic spikes and leaking corrosive acid onto the concrete, literally melting the sidewalk itself.
And we can visualize this destruction happening. The researchers use multiplex imaging.
Which is where you light up multiple things at once.
Exactly. It allows you to light up many different specific proteins in a tissue sample simultaneously so they could look directly at the liver architecture. They found that the localized toxic stress from these extruded nets severely damages all the normal healthy organ cells sitting nearby. This localized neighborhood destruction is called paracrine stress.
So what happens to those neighboring cells when they are subjected to this constant paracrine stress from the zombie nets?
The collateral damage is profound. The hepatocytes, which are the main functional cells of the liver, begin upregulating a protein called ICAM1.
What does that signify?
It's a blaring siren of severe cellular inflammation. Then the endothelial cells, which form the delicate lining of the blood vessels within the liver, upregulate VTAM1, indicating structural damage and distress.
So the functional cells and the blood vessels are both freaking out.
Exactly. And it gets worse. The stellate cells, which usually quietly store vitamin A, they undergo a transformation. They start depositing fibronectin, which lays down scar tissue, leading to fibrosis.
Wow, so the entire microscopic neighborhood is going up in flames, structurally degrading and scarring over simply because the toxic trash wasn't picked up. We have millions of these senescent neutrophils melting the sidewalks in our tissues.
That's the reality of it.
Okay, we are about 10 minutes into this deep dive. Time for another quick summary. 1. Uncleared neutrophils turn into senescent zombies marked by CXCR4. 2. They refuse to die and instead spit out toxic webs through NETosis. And 3. This localized toxic stress severely damages all surrounding organ cells.
Perfect summary.
So think about a time you felt really inflamed or achy. Could it be a microscopic traffic jam of senescent neutrophils in your tissues? It fundamentally changes how you view the aches and pains associated with getting older.
It really does.
But the massive underlying question remains here. Why do the tissue-resident macrophages, the lifelong park rangers, why do they suddenly fail to clear this toxic mess as the body ages?
Well, the researchers pinpointed the breakdown to a highly specific molecular lock and key on the surface of the macrophages. They identified a culprit, a receptor called EP2.
Okay, EP2, what is that and what triggers it?
EP2 is a membrane receptor that binds to a specific lipid messenger called prostaglandin E2, or PGE2.
Right, PGE2.
Yeah, it's a signaling molecule heavily associated with the body's inflammatory response. The critical discovery is that as these tissue-resident macrophages age, their internal expression of EP2 signaling spikes dramatically.
What does that spike do to the macrophage?
When EP2 becomes overactive, it acts like an emergency brake on the macrophage's metabolism. It drains their mitochondrial energy and severely suppresses the physical mechanics required to eat dead cells.
Okay, we talked about cellular eating, efferocytosis. That isn't just one simple action, right? It's a complex, multi-step process. How exactly does this single overactive receptor stop the entire clearance operation?
You're right. Efferocytosis requires 3 distinct, highly coordinated steps. Step 1 is recognition. The macrophage has to chemically sense and locate the dead cell.
Find the target.
Right. Step 2 is stabilization. It has to physically reach out, grab, and firmly hold on to the dead target. And step 3 is engulfment, where the membrane has to stretch around and completely swallow the cell to digest it.
Where along that 3-step assembly line does the EP2 receptor cause the breakdown?
Aging TRMs do exhibit some minor deficits in step 1, recognition. But the absolute catastrophic breakdown, the fatal flaw in the system, happens at step 2, stabilization.
So our park ranger can still kind of spot the trash on the ground, that's recognition. But their mechanical grouter tool, the device they use to actually pick the trash up, is completely broken and permanently bent. They can see the toxic waste, but they can't secure it to pull it into the garbage bag. They just keep fumbling it.
That is exactly what happens. In the cellular world, that mechanical grabber tool is a class of surface proteins called integrins.
Integrins.
Yes, and the specific integrin heavily involved here is called LFA1, which is a beta-2 integrin.
So what goes wrong with the LFA1 integrin in an aged macrophage? Does it just degrade over time?
Actually, it doesn't degrade. It gets locked in the wrong shape. Normally, an integrin rests in a folded, bent, inactive state on the cell surface.
Like a closed pocket knife.
Exactly. When the macrophage needs to eat, a coordinated team of internal coactivator proteins with names like RAC1, ROTRO-A, SCAP1, TLN2, and FERM-T3, they rush to the membrane and physically force the integrin to stand up straight.
Okay, so 5 distinct coactivators have to team up.
Right. It acts like a switchblade, opening into an extended and open conformational state. This open state creates a high-affinity, unbreakable grip on the dead neutrophil.
How does the EP2 receptor interfere with that switchblade mechanism?
Overactive EP2 signaling completely shuts down the supply chain. It directly suppresses the expression of every single one of those coactivator proteins.
Oh, wow.
Yeah, without RAC1, ROA, and the others, the integrin switchblade never opens. It stays permanently bent, folded, and completely useless. The macrophage physically cannot lock onto the senescent neutrophil.
So the ranger is standing right over the toxic trash bag, but the grabber is stuck in the folded position, and the power supply to open it has been cut completely.
And EP2's destructive influence goes even deeper than breaking the grabber. It completely rewires the genetic brain of the macrophage.
In what way?
It actively suppresses downstream genes that are necessary for digesting the cell after it's swallowed genes like JUNB and STAT3. And simultaneously, it boosts genes that actively prevent the eating process from starting at all, like CRE1.
So it's a total systemic failure of the cellular clearance machinery, all driven by this one receptor.
Exactly.
Okay, we are at the 15-minute mark. Quick recap. 1, TRMs fail because of an overactive receptor called EP2. 2, efferocytosis requires recognition, stabilization, and engulfment. And 3, EP2 prevents the stabilization integrins from opening up, meaning the TRMs literally can't grab the senescent neutrophils.
Nailed it.
So for you listening, think about this. If you knew a specific receptor was stopping your body's trash collection, wouldn't you want to turn it off? I mean, if we know this specific EP2 receptor is solely responsible for leaving these toxic zombies in the tissues, the obvious next step is finding a way to shut it down.
Which is where this deep dive moves from detailing the problem to demonstrating a really profound solution. The researchers designed interventions to turn EP2 off, and the resulting data on aging reversal is staggering.
Let's look at the clinical cases in the paper, starting with the genetic intervention. The researchers engineered a specific mouse model, the CX3CR1 Krier model, right?
Yes, that's the one.
This allowed them to selectively delete the EP2 receptor, but only in the tissue-resident macrophages, and only once the mice were already very old. When they removed that braking mechanism, did the mice physically change?
Across almost every major organ system, the mice fundamentally de-aged. Yes. Let's start with the brain. In old mice, you typically see severe, measurable cognitive decline. But when EP2 is deleted specifically in the microglia, the brain's park rangers, the aged mice suddenly regain spatial memory capabilities.
How do they test that?
They use a complex maze called a Barnes maze. And the old mice navigated it just as efficiently as young mice. They also regained episodic memory, passing novel object recognition tests that normally completely stump older animals.
That is incredible. That covers the cognitive side. But what about the physical body, like the muscle and the heart?
The physical transformations were just as dramatic. MRI scans showed a complete restoration of limb muscle volume. Furthermore, visceral fat, which is that highly dangerous inflammatory belly fat that naturally accumulates with aging, it rapidly melted away.
Wow.
And looking closely at the muscle tissue under a microscope, they observed a return of MHCII positive fibers.
What are those?
They are the specific endurance muscle fibers that give a body stamina. They normally degrade and vanish as an animal ages, but they came back.
The heart data was particularly fascinating to me. Cardiac MRIs showed that their left ventricular ejection fraction was restored, right?
Yes. That metric measures how hard and efficiently the heart pumps blood out to the body, and it was completely restored to youthful levels.
And the underlying reason for that restored heart function was visible on the TEM scans, the transmission electron microscopy.
Oh, the TEM scans were beautiful. In a normal aging old mouse, the mitochondria, the microscopic power plants inside the heart cells, they look pale, they're swollen, physically broken down, and surrounded by fibrosis.
Just exhausted.
Exactly. But in the old mice that had EP2 deleted, those same mitochondria were electron-dense. They were dark, tightly packed, highly organized, and vibrantly youthful, with zero scarring in the surrounding heart tissue.
I mean, a genetic knockout in a lab mouse is an incredible proof of concept, but we can't easily go around genetically engineering the tissues of humans who are already alive and aging. Did the researchers attempt a pharmacological approach? You know, a drug that could do the same thing?
They did. They utilized a highly selective EP2 antagonist drug known as PF04418948.
Catchy name.
Right. They administered this drug to 22-month-old mice. And to put that in perspective, a 22-month-old mouse is roughly biologically equivalent to a 70-year-old human.
Okay, so what happened to the 70-year-old mice on the drug?
The pharmacological intervention successfully unbent those integrin grabbers. By temporarily blocking the EP2 receptor with the drug, the internal coactivators flooded back, the switchblades opened, and the macrophages regained their ability to eat.
So the trash collection started back up?
Immediately. The drug massively reduced the accumulated burden of senescent neutrophils in the liver, the spleen, and the bone marrow. It cleared out the toxic trash simply by turning the machinery back on.
Which naturally leads to the most important question for anyone listening. Does this biology carry over to us? Does this exact EP2 failure and neutrophil senescence actually happen in aging human organs?
The data strongly suggests that it maps over perfectly.
Really?
Yes. The researchers cross-referenced their findings with massive, cutting-edge human single-cell databases, specifically the Gilliam's Human Liver Cell Atlas and the Tabula Sapiens project.
What's Tabula Sapiens?
It's this enormous scientific effort to molecularly map every single cell type in the human body. By pulling from these databases, they could compare the single-cell RNA transcriptomes of young human organs directly against old human organs.
And did they find the same zombie cells in broken grabbers?
The cellular patterns were identical. In aged and diseased human livers, the total number of protective tissue-resident macrophages plummeted. And in the macrophages that remained, EP2 receptor expression skyrocketed.
Just like the mice.
Just like the mice. And the uncleared neutrophils in those human organs shifted heavily into those toxic senescent NEU3 and NEU4 states we talked about earlier. The essential communication and clearance networks between the human park rangers and the cellular trash completely broke down with age.
Okay, but if this EP2 receptor, this PTGR2 gene, is acting as a master switch for immune clearance and organ decline, why hasn't it been flagged in major human longevity studies before? I mean, we have these massive genome-wide association studies, or GWAS, that scan the DNA of thousands of centenarians looking for the genes that control aging. Why would GWAS miss something this central?
Well, it comes down to the fundamental limitations of how we measure genetic influence.
Yeah.
GWAS looks at global permanent genetic variations, right? Mutations in your baseline DNA code that you were born with.
Okay.
It is a very blunt instrument. It often completely misses dynamic biological mechanisms that are highly specific to a transient cell state. EP2 isn't mutated in the DNA of aging people. The gene itself is totally fine.
Oh, I see.
The issue is that the gene's expression is being turned up too high, specifically in aged macrophages, in response to localized inflammation. A global DNA scan won't catch that state-specific localized transcriptomic shift.
Because the blueprint is fine, it's just being read too loudly in one specific room of the house.
That's a brilliant way to put it. However, if you look closely at specialized epidemiological studies, this exact EP2 receptor pathway is already heavily linked to the progression of Alzheimer's disease, various forms of cancer, and severe autoimmune disorders.
So the evidence of the damage is everywhere, even if the broad genome scans didn't flag the specific receptor initially.
Would you like?
Wow. So synthesizing everything we've uncovered from this paper today, aging is not just your cellular parts passively wearing out from use. It is a highly specific active failure of your tissue-resident macrophages, your lifelong park rangers, to clear out senescent toxic neutrophils.
Yes.
And this failure happens because an overactive receptor called EP2 chemically locks up their integrin grabbers, leaving the trash to pile up and cause toxic localized stress that literally melts the cellular neighborhood. But most importantly, whether through genetics or pharmacology, blocking EP2 proves that this biological failure is entirely reversible.
It fundamentally shifts the target. It changes how we approach the entire concept of treating aging and organ decline.
Which leaves a final, provocative thought for you to mull over. Right now, billions of dollars in anti-aging science and biotechnology are hyper-obsessed with senolytics.
Right, senolytics.
These are therapies and harsh drugs designed to hunt down and directly kill senescent cells in the body. But based on the mechanisms we've just unpacked, what if the ultimate anti-aging therapy isn't a drug that kills the bad cells? What if it's simply removing the chemical breaks from your body's own natural trash collectors, allowing your existing immune system to do the exact job it was born to do?
It suggests the potential cure for aging is already functioning inside you. The system just needs to be unpaused.
We want to thank you for going on this deeply detailed journey with us into the cellular mechanics of aging. Next time you feel a random ache or an inflammatory pain, give a little thought to your inner park rangers and consider the microscopic cleanup operation happening right now in your tissues. We invite you to return for our next deep dive, where we'll take another dense stack of research and extract the knowledge you need. Until then, stay curious.